Welcome to Unit 4 Genetics. I'm so excited to be into this content with you. We are going to be starting Concept 1 talking about DNA structure and the process of DNA replication. And these notes are more advanced. They're specifically for my honor students. So we're going to go into a bit more detail. Get excited. So just a refresher, this should be a reminder. If you're in my class from Unit 1, Concept 3 macromolecules, we learned about nucleic acids then, but now we're going to be really spending a lot of time on them this unit and the next. Remember, nucleic acids are a macromolecule that hold your genetic material. When you think of a nucleic acid, you're typically thinking of DNA. Your DNA has genes on it. These are just sections that serve as a blueprint, or you could say instructions for making proteins. And they're located at certain points on a chromosome. So there could be 1,000 genes on one chromosome. And it's so important that they have these instructions for making proteins because proteins carry out pretty much all cellular activity. They're running your cells and thus running your entire body. So it's critical that we have these instructions and that the instructions exist in every cell in your body so that your cells can make the proteins they need to function. Now, there are two types of nucleic acids. There's DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. So we'll talk about the differences in structure for both. And then we'll be talking about DNA a good bit more in this concept, but RNA will be a big player in concept 2. So first, we got to talk about nucleotides again. If you remember, this is the monomer of nucleic acids. So nucleotides make up nucleic acids, and they have 3 parts. They have a sugar, which is pictured in blue here. It's either deoxyribose in DNA or it's ribose in RNA. So that's what DNA and RNA are named after. They're named after the sugar that makes them up. There's also a phosphate group, which is pictured here in red. And then there's this nitrogen base, or some will refer to it as a nitrogenous base. But essentially, there are 5 different nitrogen bases, and then this is what makes there be 5 different nucleotides. So the nitrogen base is what's determining what the nucleotide is. So if, for example, adenine or adenine is a nitrogen base, and we refer to the whole nucleotide as adenine if it has that base on it. There's also guanine, cytosine, thymine, and uracil. Now, A, G, and C, we refer to them as the first letter often. These are in DNA and RNA, but thymine is only DNA and uracil is only RNA. So that's a really important distinction to remember. Now, DNA structure, we often credit it to James Watson and Francis Crick in 1953, their discovery of the structure, but it wasn't just them. There were a lot of people that went into it. One of those people being Chargaff, he figured out the ratio of A's always equals T's and C's always equal G's. And then I have to mention my girl Rosalind Franklin. She's the one who actually got the first images of DNA that revealed that it's a double helix. So we think of it as a twisted ladder. Here's what it would look like. Here's what it would look like untwisted. So you can see how it kind of looks like a ladder. It's a double because it's 2 strands, and then we say helix because, again, it's twisted in shape. When you look at the ladder, if you will, unwound, it has a sugar phosphate backbone. So if you notice, on the outside rung of the ladder, the sides of the ladder are alternating phosphates and sugars. And then in the middle, the rungs of the ladder are nitrogen base pairs. So those A's and T's and C's and G's, they are paired up and they're held together by really weak hydrogen bonds, which means they're easy to break. And that will be important when we get into DNA replication. All the other bonds are strong covalent bonds, which is also important because we don't want the whole DNA molecule and all the nucleotides to fall apart. We just want to be able to unzip our DNA when we need to. And we'll talk about that in a minute. So like I mentioned, these rungs are nitrogen base pairs, and they always pair with their complement. So A's always pair with T's, and C's always pair with G's. This is what Chargaff came up with or discovered. And again, these are called the complementary base pairing rules. If you like to follow United States college football, specifically in the Southeast, then I always tell my students to remember Auburn Tigers and Carolina Gamecocks. And that's how they know which go with which. Now, Our nitrogen bases, they can be divided into two types. There's purines and pyrimidines. And here's how I remember it. I think purines, that's a small word, but it's a bigger base. Adenine and guanine are my purines, and you can see them in this picture here. Notice the structure of them, how they're larger bases. Okay, that's adenine and guanine. The pyrimidines are, I think, big word, small base. So again, look at the structure of C and T. See how they're smaller than A and G, so they're a smaller base in size. And that's cytosine and thymine. And I remember which are which because pyrimidines, cytosine, and thymine, they all have Y's in them. And again, Chargaff was the one who figured out the A is always equal T's and the C is always equal G. So we always have one bigger base and one smaller base that form hydrogen bonds. And again, these are weak bonds. If you notice in the picture, you'll see also that A and T form a double bond and C and G form a triple bond when they base pair. So that's important to note. Now, something you may have noticed from all the way back here is how we have a phosphate sticking up on this end but not here, and then how these look like they're upside down. That is because DNA is considered to be antiparallel. That means the strands run in opposite directions from each other. One of the strands runs in a 5' to 3' direction. That's just what it's called. And then the other strand runs in a 3' to 5' direction. Now, how do you remember this? Phosphate end is always the 5' end. So I enunciated that way on purpose. Think phosphate 5. So this end with the phosphate sticking out, those are your 5' ends, and then the deoxyribose side, that's the sugar side. So that's how you can always look at a picture and figure out which strand is which. Okay, so notice they're running in these opposite directions. That's important. Okay, so let's practice labeling this, okay? So first, let's find our 3' and our 5' end. Always figure out your directions. So remember, 5' phosphate. So look for the little phosphate groups sticking out. Those are your 5s. That means the others with the sugars are your 3s. So this is my 3 to 5. This is my 5 to 3 strand. Now, what's that again? That's your phosphate. That, remember the alternating, it's a sugar phosphate backbone. So phosphate, sugar, phosphate, sugar, phosphate, sugar. So that's our sugar. It's DNA. So it's specifically deoxyribose. And then remember I said these are held together by strong covalent bonds. In the middle here, okay, we've got our nitrogen bases. Now these arrows are pointing to the larger bases. So are those purines or pyrimidines? Well, remember, big base, small word. So if those are the bigger bases, those must be purines, which are adenine or guanine. My littler bases, little base, big word, that's my pyrimidines. And remember with the Y, so that's either cytosine or thymine if I was going to label those. And then what kind of bond holds those together? A weak hydrogen bond so that I can easily unzip my DNA, which we're going to see in a minute. And then this entire thing, that phosphate, sugar, and nitrogen base, that's a nucleotide, which is the monomer of nucleic acids. Now, those nucleotides make up RNA too, but it's a slightly different overall structure. RNA is just a single strand of nucleotides with exposed bases, so they're not paired up. But there are times we're going to see where RNA bases will pair up with DNA and vice versa. And when that happens, A's go with U's and C's go with G's. Because remember, RNA does not have thymine and DNA does not have uracil. So that's important. But if you're using our college football memory tool, it still works. Auburn Tigers for DNA and Auburn University for RNA. And then it's always Carolina Gamecocks. And if y'all follow me or know me at all, you know I'm a Clemson fan, so it's hard for me to talk about the Carolina Gamecocks, but I will do it for the sake of memory. All right, so let's just summarize an overall chart to compare and contrast their structures. Okay, so when we're thinking of DNA versus RNA, one of the main distinctions is the types of nitrogen bases. They both have AGC, but DNA has thymine and RNA has uracil. That's super important. The type of sugar is what they're named after. So deoxyribose is for deoxyribonucleic acid and ribose for ribonucleic acid. And then if you were going to summarize DNA structure and its shape into 2 words, most would describe it as a double helix, and then RNA would just be a single strand. Now, these are some terms we've mentioned before when we learned about mitosis, but I need to bring them up again because it's so important you have them straight. Oftentimes, people say genes and chromosomes and DNA interchangeably, and that's not always entirely false, but it's not always completely accurate. So I want you to know the differences between them. So your chromosomes are tightly coiled strands of DNA. Your DNA is very long, and so it has to be really tightly coiled up to fit into your tiny cells. And different organisms, their DNA is organized into different numbers of chromosomes. So for example, humans, we have 46 chromosomes in each of our body cells, or you can refer to it as 23 pairs, because one in each of those pairs came from your mom or your biological mother in the egg, and then one came from your biological father in his sperm. So that's kind of where that number comes from. Dogs have 37 pairs or 74 total. So it just depends on the type of organism. It's doesn't necessarily mean too much. Okay, now a gene is a section of DNA. Thus, it could be a section of a chromosome that has the instructions to code for a protein. One chromosome can have thousands of genes linked together on it. So if I was going to summarize this into simple statements, genes are pieces or sections of DNA. Chromosomes are long strands of DNA all bunched up. And I like this image. It's not perfect, but I do like how it kind of helps us visualize this. So look here at our DNA. We see our nitrogen base pairs, our A's with our T's and C's with our G's. Notice how it's getting coiled up here. We can also see histones are these proteins that they wrap around. They form nucleosomes, and that's what makes the tightly coiled chromosome shape. And then you can see the chromosomes in the nucleus of the cell. So I really like this visual because just a section of this could be a gene, or a section of this part could be considered a gene. Now, the one thing I don't like is, remember, this is so important. When a chromosome looks like an X, those are sister chromatids. Those are duplicated chromosomes. So 2 copies of the exact same chromosome. So this is just showing chromosomes after the S phase of interphase. So before cell divisions happened, but after synthesis, and that's important. And now we're going to learn about that S phase. So if you remember when we did mitosis, we said before the cell divides, it's got to go through G1, S, and G2. In S phase of synthesis, we're going to double our DNA. Today, we learn what that process is, and that process is DNA replication. So when a cell is ready to divide, it's going to first copy its DNA. And this process of making an identical copy of DNA is called DNA replication. DNA making more DNA. And this happens in the nucleus because that's where your DNA is located, and it happens during S phase of interphase. And this is so critical. It's ensuring that each new cell that's made from cell division has the same DNA as the original cell. So think about it. Let's say your hair cells are dividing, which they do. One of your hair cells has 46 chromosomes in it. If it never doubled its DNA, when it would divide and make 2 daughter cells, each one of those would only have 23 chromosomes. They'd only have half of your DNA in them. So they could only have the instructions for half of the proteins. And then if those divide, then they each only have 11.5. Okay, or and vice versa. I think that's the right mental math there. So it's so important that instead, before the cell divides, S phase happens, we double our DNA, so we have 2 copies of all 46 chromosomes, or 92 individual chromosomes, or chromatids, and then those will divide so that each resulting daughter cell has 46. That's super important. Okay, so how does this happen? This process is mediated entirely by enzymes, like pretty much every process in your body is. And enzymes are proteins, don't forget that. So we're going to walk through each step, and I'll highlight all the key enzymes that I want you to know in purple. So first, enzyme helicase unzips the DNA into 2 separate strands. So remember we said these are weak hydrogen bonds holding those nitrogen base pairs together? We're going to unzip those thanks to helicase. And it does this, it opens up at something called the origins of replication. And this will happen at several places along the DNA at once. So think about a rain jacket. If you're going to unzip a rain jacket, you start from the top and you unzip it all the way down to the bottom. That is not how this works. Your DNA is way too long. Instead, it would be like if you started pulling apart the zipper on your rain jacket at multiple points along the zipper, not just top to bottom. That's what's happening here. So we're going from this to unzipping it like this. Now, step 2. An enzyme called DNA polymerase is going to add complementary nucleotides to the template strands. So remember those complementary base pairing rules. A's go with T's and C's go with G's. So all these places where I see an A, a T is going to come. Where I see a T, an A is going to come. Where I see a C, a G is going to come. And where I see a G, a C is going to come. Now, here's something that's important. DNA polymerase only works in certain directions. It only adds nucleotides to the free 3' end of the template strand. So it's making its new DNA strand only in the 5 to 3 direction. So watch this. It's adding on this side and this side and this side and this side. So notice how it's adding them in that direction. We're making a 5 to 3 strand regardless of which side we're going. Now, this whole step is especially important that we have all the enzymes. So there's a couple of them in here, not just DNA polymerase like I already mentioned. First is primase. Primase is required for DNA synthesis. I like to think of it as a key for your car to turn on. You've got to have it so that your DNA polymerase knows where to get going. So what primase does is it makes a very short RNA primer, which is basically just a really short strand of RNA that tells the DNA polymerase, hey, come here and now make DNA. So that DNA polymerase does a lot of things. First, it adds nucleotides to the RNA primer. So this is like its signal. It goes to there and it starts adding the nucleotides. Then once it's done that, it goes back and it removes the original RNA primers with DNA. So that's the other thing it does. And then it also is our proofreader. So it goes back and makes sure, okay, did I get all the A's with the T's and all the C's with the G's? Because if you mess this up, remember, you're messing up the instructions for making a protein, and proteins run your show. So it's really important that we copy the instructions correctly. And then another critical enzyme is ligase. It's going to seal the gaps in the DNA and make sure everything is connected well. All right, so here's a picture of it. We've got our enzyme helicase pictured here, and it's the one that's unzipping our DNA. We have primase, which is adding RNA primers so that DNA polymerase knows where to get started. DNA polymerase here is adding things. It's only creating a 5 to 3 strand. So notice it's moving in this direction on this strand and this direction on that strand. And that's super important. And then ligase is coming through and it's sealing everything back up together. Now, notice in this picture, I reference a lagging strand and a leading strand. Because it can only work in certain directions, one strand is made really smoothly and one is kind of made in a funky way. So first we have our leading strand, pictured here. This is a new strand that's made toward the replication fork. So like as we're unzipping, we're also building new DNA in the same direction. Remember, we're only making DNA in the 5 to 3 direction because we're using the 3 to 5 side as a template. Because of this, because it's going in the direction that we're unzipping, we only need one primer to get started, and then it's going to be able to just replicate continuously until we get to the end. The lagging strand is honestly just, it feels very inefficient, but essentially we're making DNA away from the replication fork because we can only make it in a certain way. And so because of this, it's going to have to replicate discontinuously. And it makes these fragments of DNA that we call Okazaki fragments, which are just short pieces of DNA. And then ligase is going to have to go back and seal those together. And it's going to require a lot of primers so that it'll keep doing this process. And this sounds a lot more complicated than I think it is. I have a really great animation I'll show you in class that'll help you visualize that a little bit better. That's not so, as stagnant as a picture. Okay, once we get all of our A's and our T's and our C's and our G's together, we should end up with 2 identical DNA molecules formed, each that are half old and half new. Because remember, one side came from the original DNA, and then we brought in new nucleotides for the other sides. So that's why we call replication a semi-conservative process. So look at this blue little DNA ladder. Consider this the parent DNA. Each parent strand, when it unzips, becomes a template or a pattern that determines the order of the new bases. So the complementary bases that come in match with the original strand. So our end result is two identical DNA to the original. They're half old, which is the blue, and then half new, which is pictured in red here. That's what we mean by semi-conservative. So again, the overall process, we're unzipping DNA, we're using in a bunch of enzymes to a bunch of things, but basically they're adding A's with T's and C's with G's, and then we end up with two identical strands that are half old and half new. Now, a couple of things I want you to consider. What kind of mistakes could happen? There are a lot. What would be the implications of those kinds of mistakes if we do some part of this process wrong? And consider, okay, what would be different about a mistake happening when we're doing DNA replication in a brain cell versus if we're doing DNA replication in a germ cell that's going to end up making a sex cell like sperm? And what would be the difference if a mistake was happening in a gene versus an entire chromosome? These are really critical questions that we're going to talk about in class, and then we'll spend a lot more time on them in Unit 5 Heredity. But they are things I want you to consider because this is really important. And that is DNA structure and replication.